Good morning, everyone. Welcome. Our speaker today is Dr. Akhilesh Kumar Verma, who is a postdoc in civil and architectural engineering at the University of Miami, working under the supervision of Professor Luis Ruiz Pestana. Dr. Verma received his MS from the School of Physical Sciences at Jawaharlal Nehru University, New Delhi. Then he completed his PhD under the supervision of Professor Rahul Pandit at the Department of Physics, IISc Bangalore. He then pursued his first postdoc with Professor Dwight Barkley at the Warwick Mathematical Institute in UK. And of course, he's doing a second postdoc right now with Louise. His research interests include biophysics, soft and active matter, fluid and superfluid turbulence, and self-gravitating systems. Today, he's going to talk to us about emergent behaviors and mechanics in biofilms and active solids. Before he starts, Louise will say a few words.
Yeah, thanks, Pranay. I just wanted to say that what we are doing is not conventional civil engineering, but I see it as the next frontiers in the stuff that we care about. And Achilles is sort of the right person to be working on this because he comes with a very different perspective. He's a physicist. He wasn't working on this stuff before he came to my group. I wasn't working on this stuff. So this is something that we have been sort of growing together doing. And it's been very exciting. So yeah, I'm glad that Achilles got the chance to tell everyone what we have been doing for the last 2 or 3 years. So Achilles, the first.
Thanks, Pranay, for this nice introduction, and Louis for the description about our work. And first, I will thank Pranay also for giving me opportunity to present our work, what we are doing here from last few years. So I'm going to talk about the emergent behaviors and mechanics in biofilms and active solids. And I will introduce as we go along what I mean by emergent behaviors. And most of the audience here are the civil engineers, then they know very well what mechanics I mean. And I will talk about the biofilms and active solids in great details as we go along. So Pranay has already told me about myself, so I will not repeat those things again. Just I want to say that what I'm going to present is just I'm working with Louis from last few years, and these all works done here in University of Miami. So what generally civil engineers do? So they study and build the great structures like Eiffel Tower, Golden Bridge, giant this building called Burj Khalifa, and great dams like Hoover Dam. So if you notice that in this all structure, one thing is common, and it is that these all structures are passive. Passive in the sense that they do not consume energy internally, and they only deform or move in the presence of some external force or interaction with the external environment. But there is another set of material, a system called active system, and these systems are like next frontiers in mechanics. And the example of such kind of systems are like biofilm. And biofilm is basically a community of bacteria, which we'll talk in great details later. And then another example of active system is tissue. It's just basically the collection of cells. And synthetic example, these two, biofilm and tissue, are the naturally example of active systems. And we can have a active solid, which is basically the synthetic active system. And here you see the common in this all system is that their basic building block is active. So for example, bacteria's building block is basically the bacterial cells. Here the tissue formed by the collection of active cells. So if you see in these all systems, what happened that these basic building blocks, they start doing consume energy internally, and they start producing some motion. And their motion leads to a global behavior, emergent phenomena. And these emergent phenomena are very different. And it basically arises from the interaction of many degrees of freedom among the active units. And these behaviors are also very different from the behavior of its basic building block. So for example, here in this biofilm, if you see a single bacteria, active bacteria cell, then it's very hard to predict that if you put many together bacteria, then they can wrinkle. But eventually, if they form their community, then they wrinkles. And the wrinkle is here the emergent phenomena, emergent behavior basically. Similarly, in this tissue and active solid also, they start producing some collective motion, and this collective motion are basically the emergent behavior. So in this presentation, I'm going to talk about the mechanics and emergent behavior in these two kind of system, like one biofilm and active solids, and another is active solid. So this is going to be the roadmap of my talk today. First, I will talk about the biofilm. And in this presentation, I will talk about the role of friction, adhesion, and nutrients on the biofilm mechanics and morphology. But our grand goal of this project is to understand the role of phenotypic heterogeneity on the biofilm formation, how the phenotypic heterogeneity affect and lead to the biofilm formation and its wrinkling behavior and those all things. And this work is funded by HFSP, and the principal collaborators of this project is, of course, Dr. Luis Ruiz Pestana. And along with me, we are just like developing the model in this project, and Diana Fusco and Carolina Tropini, these other two groups, one from the Cambridge and another from the Universities of British Columbia, Canada, they are doing the experiment. And we are working together to understand this biofilms and understand this phenotypic heterogeneity and how it controls the wrinkle formation and biofilm formation. And in the second part of this presentation, I'm going to talk about the active solids. And in this active solids, we are going to see the different emergent phases and its properties. And we are working on this project with Olivier from the CNRS Paris. And actually, it's very interesting story how we start working on this project. We didn't think about this project until we start working on this. And actually, I was studying, just Google searching some literature survey, and I found a paper which appeared in Nature Physics few years back. And I read it, it's very interesting and tempting. Tempting in the sense, two sense. First is that it is a very new area of active research. It's just like in few years, it has like just people are getting involved in that project. And second is we had developed all the tools which is required to solve this problem when we were working on the biofilm. Just because of the two reasons, I got really interested, and I just tried to reproduce those results in that paper, and we got something new. And I saw it to Louis, and he also got interested, and that's how we start working on this problem. So in the second part, we'll talk about this active solid. So first, what is the bacterial biofilm? So bacterial biofilms are basically communities of bacteria microorganisms that grow at the surfaces. And how they form? Then consider the motile cell. Motile cells are basically cells which can move independently and they can swim. But what happened that when these cells swims, then due to lack of nutrients, they have some stochastic switch in. So they change their phenotype. And when they change their phenotype, then they turn from motile cell to matrix producing cell. And this matrix producing cell has a capability to release some extracellular matrix. And this matrix has elastic in nature. So what happened that these cells, motile cells, and matrix producing cells, they start getting stick together due to the presence of this extracellular matrix. And that's how the system, these bacterial cells build their community called the biofilm. So where we can find the biofilms? So we can biofilms are almost everywhere. And one example here I'm showing is the example of biofilms in the Yellowstone National Park. And here you can see this yellow color kind of stream, and which is just filled with the giant biofilm. And another example you can find the biofilm is in this water pipe. What happened that when some bacteria stick to the internal surface of this pipe, and they form their colony, and eventually they damage the pipe. And we can find this biofilm inside our body also. For example, we can find in the lungs, we can find in our guts, and biofilm can grow on our skin also. Here we see showing that some bacteria can stick to our skin, and they start growing their community, and eventually they damage it. And it has been estimated that roughly we lose $5 trillion globally due to the biofilm. And imagine this $5 trillion is just like 20 to 25% GDP of the US. That's very huge money. So to understand the biofilm, we need to understand this, how it wrinkles. So I'm going to show here a cartoon. So what I'm going, here you can see that this color is showing the substrate, and the green color is a bacterial community. And what happened that from the substrate, this bacterial community eat and they grow. And when they grow, then they just try to relax. And this relaxation has been like resisted by the friction. And due to that, some compressive stress start building in the system. And if you add the addition, then what happened that addition is basically in the interaction between substrate and biofilm. Then this compressive stress just also get amplified, and biofilm eventually wrinkle. And here you see the example of a wrinkled biofilm. Here you see this in the center you see small wrinkles, and these wrinkles are like randomly oriented. But if you see these big wrinkles are like spread throughout in the radial direction. So what is the importance of wrinkles in biofilm? So wrinkles are important because they flow the nutrient and oxygen, and it helps to give the food to the bacteria which are like not able to get. And they can use this as a waste removal to survive for longer. So they can, and this biofilm also use this wrinkle as a micro environment to protect them from the antibiotic. And they can use also to colonize some tissue and medical plants using this wrinkle. So basically, this bacterial community use this wrinkle to enhance their fitness and survive longer. So to understand this all wrinkling behavior, what we do? We go and do the experiment in lab. So here I'm going to show you a few snapshots from our collaborators. So these snapshots are taken from their experiment. And what you see, this green color is the motile cells, and this pink color is matrix producing cell. So what happened that initially if we put on the they put motile cells on the substrate, and substrate is basically contains nutrients, and bacterial cells eat those nutrients and grow. And after a while, due to some lack of nutrients, other stochastic switch, they produce matrix, and this matrix wrinkles. This matrix helps biofilm to wrinkle. So initially they wrinkle in the center, and when it builds the enough stresses, and eventually they start wrinkles start propagating radially, and eventually they form a mature biofilm. And here you see that there is a wrinkle structure in the biofilm, and there you can see that there is a heterogeneity in the phenotypic expression also. Phenotypic expression, I mean the distribution of matrix and motile cell also. So our aim is to develop a model which can help us to understand this spatial temporal behavior of phenotypic expression, wrinkles, overall of the biofilm. And in experiments, it's very difficult to control this all phenotypic expression and this all thing. But if you do simulate it simulate it, then we can easily do. So simulation has that advantage, which we are, it is very difficult to do those things in experiment. So we develop a model. So what are the basic building block of our model? So what happen in our model, there is a nutrient availability. So biofilm eat those nutrients and they produce biomass. And then we put the constitutive behavior and substrate interaction, which lead to give biofilm morphology. So here we consider in the constitutive behavior, we consider the system behave like a linear elastic, and the properties of the elastic behavior is uniform, and we consider like a solid. And in the substrate interaction, we consider that the biofilm interact with the substrate using the addition and friction. And here, if you see the transparent gray color, here we have, this is work on progress. And here we have not, today I'm not going to talk about this phenotypic expression, ECM production and phenotypic pattern. But if you see that this all like nutrient availability, biomass production, constitutive behavior, and substrate interaction, these all are the local properties of the biofilm. But if they all acting together, then they eventually give the emergent behavior, and that's the morphology. So now we are going to build our model. So what we do that we take a substrate, and substrate basically contains the nutrient. So we discretize the substrate in square lattice form to model the nutrients evolution on this substrate. And for the biofilm, what we do that we coarse grain in hexagonal patches. And the properties of this hexagonal patches we represent at the center of the hexagon. And to model the elastic properties, which is arise in the biofilm because of this ECM, we connect the center of the hexagonal patches by spring. And thus, this biofilm is basically the spring network, triangular spin network. And the distance between two center of the hexagonal patches is L0, and this L0 is the smallest length scale in our system, and we can't say anything which is happening below this length scale. So this is the smallest length scale in our system. So what happened that if you put the nutrients, then biofilm eat those nutrients and they diffuse. So we model the nutrients evolution using this reaction diffusion equation. And this is basically the diffusion equation, and here the reaction term, you can say this uptake term. So basically biofilm eat those nutrients and grow. So here we model the growth of this biofilm patch by increasing the size of this biofilm hexagonal patch, and it is modeled by using the Monod's law. And when this biofilm grow, then what will these are the connection of springs, so they will generate the force. So to model that force, what we do that we model the in-plane interaction using this potential energy given by kV by this harmonic potential energy, and this these bonds form improper, and then we model the out of plane interaction using this again harmonic potential, and here the biofilm can attach to the substrate also. So this substrate interaction we model using this adhesive potential, and here we this adhesive potential is basically the linear we use the superposition of the Lennard zone potential and Yukawa potential. So here we show the cartoon of the biofilm interaction with the substrate, and the friction and interaction among the nodes. So here you see that this this node is attached to the substrate, and when it tried to move, then it is resisted by the friction due to the substrate. So we model the when the biofilm evolve, so we evolve using this Langevin dynamics, overdamped Langevin dynamics, and here dV is model the friction. This is the friction between substrate and biofilm, And you see that here dB is inversely proportional to friction. Here zeta is the friction. So higher friction is corresponding to lower dB and vice versa. So if you solve these equations, then we get the biofilm. So what initially we solve, okay, so we solve this equation of motion along with this other equation which I show on this slides. So we get the biofilm dynamics. So here I'm going to show you biofilm formation under the constant nutrient supply. So here what do we mean by constant nutrient supply? Here we consider that the nutrient are distributed remains constant throughout the space and time when the biofilm growing. So what basically we are doing, we are like complete there's infinite nutrients, so biofilm has can eat and grow, and there will be no deficiency in the nutrient concentration. So when this so here I'm going to show you how it happens. So initially they eat and grow, and what happened that in the latter they wrinkle in the center, and they form some blister-like kind of structure, and then they again coalesce, and they eventually form a radial wrinkle. So why they wrinkle first in the center? So to understand that, what we do that we plot the radial stress profile just before the wrinkling, and what we observe that biofilm has built the large stress near the center, and it has less stress near the periphery. And why it happens so? Because periphery has free space to expand outwardly, but the center is highly confined, and hence it build the high stresses in the center. And this lead to biofilm wrinkle in the center. And our collaborators, so here I'm going to show you some experimental results from our collaborators. And here they use the finite nutrient concentration. Earlier we saw the infinite, but here they use finite. And what they show that if you use finite nutrient concentration, then if you use low nutrient concentration, then the biofilm first wrinkle near the periphery. And here you see this is the time of wrinkle, the first wrinkles near the periphery. And if you increase the nutrient concentration, go at high nutrient concentration, the first wrinkle in the center. So we see two different kind of wrinkling mode. So we ask the question, can our model able to capture these two kind of different behavior? So here we so that at low nutrient concentration, we find that as first mode appear, where the biofilm wrinkle first near the periphery. And if you increase the concentration, then biofilm can wrinkle in the center, just like what we observed in the experiment. So we have already understood why it first appear a wrinkle in the center at high nutrient. To understand the nutrients, to understand why it wrinkles first near the periphery, we plot the stress nutrient concentration profile. Here I show just two different snapshots. And here you see at early time, it is just like wrinkles at the periphery, and then the wrinkles start moving inwardly. And here I'm going to show the nutrient concentration profile. So what we observe here, that initially at time t equal to 0, nutrient concentration is uniform. And as time grows, what happens that biofilm eat the nutrients in the center, and nutrients get depleted here. And around the periphery, there is always plenty of nutrients, and which they eat and grow. And in that process, what they do, that they build large stresses. You see here the large stresses, circumferential stresses near the periphery, and which leads to this radial wrinkle. So basically, due to lack of nutrients in the center, biofilm doesn't grow much, and in the periphery they grow, and they build the circumferential stresses, which lead to the radial wrinkle near the periphery. So we see these two different kind of wrinkling mode. So we ask the question that can we understand the nature of the transition, how the transition happen. So here we calculate some order parameter, and this order parameter is defined following. We calculate the location where the wrinkles appear from the center, and if it is the size of radius then of the biofilm, then it will be 1, and when if it wrinkles at the center, then it will be 0. So we continuously change the nutrient concentration, and we see that first it wrinkle near the periphery, and then eventually slowly it just going to in the and then it wrinkles in the middle of the biofilm, and eventually it wrinkle at the center. So it more seems like a continuous transition. So we also study the effect of friction on the wrinkling mode, and we find that if you choose nutrient concentration not very high and not very low enough, then at high friction the biofilm wrinkles first in the center. But if we reduce the friction, then what we found that it can wrinkle near the periphery also, and the mechanism is exactly the same what we explained for the concentration case also. And this behavior is also has been observed in the experiment. So we see that just playing with the nutrient concentration, the friction, we can get two different wrinkling modes. So we ask the question that can we get some phase diagram kind of can we get some phase diagram to for this edge first and center first transition. So here I'm going to show you a phase diagram. So here we choose the addition, here we keep the addition constant, and here we are changing two parameters. One is the initial concentration, nutrient concentration, and then we are playing with this friction. The here friction, higher friction is corresponding to higher dB is corresponding to small friction value, and vice versa. So here we observe that for very high nutrient concentration and low friction, biofilm wrinkle first near the center, but as we reduce the nutrient concentration, then it can it can go to the edge first mode, and we see just kind of like a stepwise kind of function which separate this two phase diagram, two phases. And we also study the this phase transition between the we fix the friction and we play with the addition, the interaction between biofilm and substrate along the z direction, and we see that they have this two parameter space has also similar kind of phase diagram. Okay, at higher nutrient concentration and higher friction, they show kind of center first, but if you reduce the nutrient concentration, then it can transit to the edge first mode. So what are the implications of two different wrinkling modes? So we see that at higher nutrient concentration, we get the wrinkling mode at the center. So biofilm, it does possibly to felicitate the cells, dispersal of the cells which are like in the center to colonize the other new environment just by going outward. So it creates wrinkles so that in the center, so that the bacteria or the cells which are inside, they can go out and they just like grow their own community to somewhere else in the new environment. And the advantage of having these wrinkling modes like this circumferential wrinkles can provide the food to the bacterial cells which are starved in the center. So this wrinkling mode can let the nutrients and oxygen supply from outwards and come inside and feed those starved cells which are stuck at the center. So this two different wrinkling mode has huge fitness advantage, used as a fitness advantage for their survival and growth. So now we are trying to add in our existing model some viscous relaxation and different phenotypes to study the complete and more realistic model for the biofilm. And here we are adding different phenotypes by means like adding the some cell matrix and motile cells. In this study, we have assumed that all cells are like matrix producer cells. Hence, we do not see those phenotypic switch and those all things. But now we are incorporating the phenotypic expression in our model, and this is ongoing work. So with that, I end the discussion on the biofilm here. And now I'll move to the second part of the presentation, that is the active solids. So I'm going to show you a movie which is taken from this paper. And this is a movie of, again, a bacterial biofilm, but this is a very different kind of bacteria. What earlier I saw, it was like B. subtilis and E. coli. But this is a different bacteria, and when it form a biofilm, then it has kind of crystalline kind of structure, and it has been shown from the SCM image. Then they form the crystalline kind of structure. So here I'm going to play the movie, and they have grown it in some circular disc environment, and this bacterial biofilm is this and since it is a crystalline form and it's active, so it is a good candidate of active solids. So here and this bacteria, this biofilm is light sensitive. So you see that when light is off, then it is showing some collective oscillations. You can see they are moving synchronized. They are in the sync, and they are moving kind of anti-clockwise. But what happens when you turn on the light, then their activity start reducing. If you see the activity, then it start reducing. And then they start changing their motion. And from collective oscillations, what they happen that they start showing some global alternate rotation. First they are like clockwise, then turn in the anti-clockwise, and then they are repeating this clockwise, anti-clockwise motion. They are doing that. So this bacterial, so this is example of active solids. So this active solid showing two different kind of collective behavior, just depend upon the light. And here the light basically control its activity. So just by playing with the activity, you can get two different kind of collective behaviors. And this is a natural active solid system. So now what I'm going to show you, I'm going to show you another active solid system, which is more kind of synthetic. And here what you are seeing that this is a hexabug, and this hexabug has some motor which gives this create energy to move. And they connected with this like 6 hexabug in this hexagonal form. And when they turn on, then you see that initially it is okay. So they are controlling the tension in this structure by this extended object. And initially at the low tension, what we see that global alternate rotation. They are just like going clockwise and anti-clockwise. And slowly what they do that they are just like going to increase the tension, and they have increased, and see that they start showing some global behavior, global collective oscillations. So we see a completely a similar kind of emergent behavior in two completely different systems. One is a very natural system, and one is synthetic system. So these are the example of the active solid system, and these are the kind of collective or emergent behavior in this kind of system. And you see these collective motions. So why we need to study the active solids? So I'm going to show you another movie which I took it from the YouTube. And the main purpose of showing this movie that active solid system can be helpful to mimic the motion of different species. And here I'm going to show you this movie. I'll see the motion of some species. Here they are more kind of crawling. So you see here robot is moving in the uneven terrain and it collapses. But if you use some active solid system, then they can like navigate the unusual terrain. And here you can see that they are doing kind of some rolling motion. But if you change the see here, they are doing the rolling motion, but if you change the environment, then they can adopt it and they start doing some crawling motion. So basically, active solids can be useful to understand the motion of different kind of species, and they have other advantage also that they can be used in biological system and medicine. For example, wound healing, art, making some artificial tissue and organ, and they can be also used as smart materials, which can adapt to stresses. It can heal cracks and change shapes depend upon the environment, and they can also use as soft robotics because those hard robots are like sometimes not safe to interact with humans because they can injure human. So soft robotics can be active cells can be used in soft robotics. So now the question is how we can model this kind of system. So here I'm going to show you that just like take how we build the active solid system in model. So here, just like if you take if you remove this polarity, this red color vector arrow, then what we see that it will be just simple solid. So to model the active solid, what we do that we put the each unit as a active unit and the activity has its own polarity which is given by this vector n. So to understand this, what you do, you just write the equation of motion. So here in the first equation of motion, this is basically the Newton equation. And here you see this F naught is basically the activity which is coming from the, which is generated by the each node due to its internal mechanism. And here gamma V is basically the a damping term. Is just coming from the environment in which this material our object is like studying are situated. And this is basically the spring force on any particular node. And just sum of the all its nearest neighbor. And it has some extra degrees of freedom, this n, which is coming from the interaction just because of the activity. And this n is its equation of motion is given by this equation. And this equation is basically trying to align this polarity along the velocity direction. Okay, and here this is just like some thermal noise coming from the environment. So from this system, you can get some dimensionless parameter pi, which is basically proportional to activity. So if you increase the activity, basically you are increasing the pi. So we ask the question that if you solve these equations, and then how it is going to show the motion. So what we do in our simulation that we fix the edge, we pin the edge. So whatever the boundary we see here, we pin them so that it is not showing some trivial solution. And then what we see that if you pin the edges and solve those equations, so what we found is this. Here, I want to tell you that this is, I am plotting the velocity vector. Velocity vector is just basically V. So it's, the length of arrow is showing its magnitude and the arrow sign is showing the direction. So at the very low activity, what we see that it is almost frozen and there is just small fluctuations in the system. So activity is not high enough to make the spring compress or stretch, and so that it can generate some motion. So it is just like there is some very small fluctuation, very tiny fluctuation. So basically it is just like not doing anything. But if you go to higher activity, then what we see that, for example, here, that we see that system start oscillating. And you can see that they are doing this all collective oscillations. And these all basic units are in the same phase. They are moving in the very synchronized way, and they are creating some collective oscillation. So this study has been already reported. But what we found even more interesting is that if you go at some higher activity, for example, at pi equal to 0.2, 2 times compared to previous one, then what we found that we find some topological defects. So here you see that again, it is showing some collective motion, but this has a very different properties of phase than what we saw. Here it form a vortex, and this vortex move and it in there, and again it's recreate and move. So it is just like a kind of vortex which is propagating throughout the system. And this happens just because when you increase the activity, then the polarity and velocity, they are not able to, there is a phase lag between them, and because of that, we see this topological defect. And even if you go on some even much higher activity, for example, pi equal to 3, then what we observe that system goes from, it form a vortex, form defect, but it become highly chaotic. It is not like it's become very highly chaotic system. So we see different kind of phases on changing the activity. We see different kind of collective motions. Okay. So now we ask the question, can we understand this behavior? So can we describe this at least can we come up with some order parameter which can characterize the different phases? Then we calculate this topological charge. Is basically the just like is taking calculating some angle around some it's just basically calculating the angle around a closed loop, the difference in the angle around the closed loop. And here we see that for when all the phases are like align in the same direction, then this topological charge is 0. But when it form a vortex, then it has topological charge 1. And here we see that this vortex like get expelled or absorbed near the boundary. So here there is no defect in bulk, and hence we get the topological charge 0. And if you go at the higher activity, then we get some higher topological charge, and this topological charge nothing but tells about the number of vortex. So we also try to calculate the spatial correlation function. So here we calculate the spatial correlation function of this angle theta between the velocity. And here we plot it for the different phases. So here we see that at very low activity, when they all doing this collective oscillations and being in the same phase, then what we see that this spatial correlation function almost remains constant. But if we increase the activity, and then what we see that it form a defect, but this defect are not in bulk and it get absorbed at the boundary. And so in this case, what we see that the correlation start decreasing, but it remains get almost fluctuate around some mean value. And it's just because you can see some parallel structure after some certain length scale. And if we increase, and here we see the correlation function for this defect. So here we see that its correlation function decreasing. And after a while, we see that they become anti-parallel, and hence they become negatively correlated. And at very high activity is just, we see exponentially a decaying function in the correlation just because it's become the chaotic system. So these different phases has been characterized by this spatial correlation function of the angle between the velocity. And with that, I would like to, in my talk, just giving some key takeaways. And from the biofilms, what we develop a computational model that can independently control the addition and friction. And we found that just playing with the nutrient concentration, we can get two different wrinkling modes. And these wrinkle modes are like helpful for the biofilm for their fitness advantage. And in active solids, we observe that we find a new phase on increasing the activity, and it has topological defects. And if you increase more activity, go even on higher activity, then we find that this topological defect proliferate and it turn it into the chaotic motion. So with that, I would like to finish my talk, and thanks my PhD advisor, of course, Louis. And it is, I thank him for his guidance and support. And it is really, I enjoy working with him. And his passion for science and enthusiasm for science is really inspiring. And these are my collaborators with whom I am working on. So yeah, thank you very much for your attention, and happy to take few questions, many questions as well.